Hello, everyone. Welcome to my video tutorials for single-cell RNA sequencing data analysis using the Giotto package. In today's video tutorial, I'm going to show you how to perform data integration in Giotto. So first, let's note the package Giotto and also tidyverse. For today's demonstration, I'm going to use two datasets from GSE 132771 publicly available dataset. So the first one is the sequencing data from healthy human lung. They are purified mesenchymal cells. First, let's create a Giotto object for the healthy human lung. Let's run. So you can see in the data window, we have a AML Giotto object for the healthy human lung mesenchymal cells. Then next, we can create a Giotto object for the fibrotic lung, and we name it as IPF. So now you can see we also have the IPF Giotto object. So we are going to use AML and IPF Giotto objects and perform data integration. So first we need to join both objects. This is a very similar function like the merge function in the thread package. To use the joint object function, you can see we have the Giotto object list, AML and IPF. We can give a name as AML and IPF as well. Then the joint method will be Z-stack. So we can join both objects as AML-IPF. Let's join both objects and create one joint Giotto object. We can run. So you can see now we have a joint object to contain both normal sequencing data and IPF sequencing data. So in the first step, we created a joint object for data integration. If you have more than two datasets, you can just add the object names into the list. So next step, we can perform the filtering to get rid of the low quality cells. I introduced those functions in the standard workflow, so we can use expression threshold as one, keep features that detected at least in 3 cells, and we keep cells that have at least 200 gene expression. Then we can use the criteria we set here to filter the dataset. Let's run again. So you can see down here, we removed 4 cells from 13,614 cells. And also we removed 2,872 genes. In the beginning, we have 22,404 genes in the joined Giotto object. So after the filtering, we can normalize the data. Let's run again. So following the data normalization, we can add the mitochondria gene percentage into the object. So first we need to get the mitochondria genes from the object. We know the gene names are in the feature ID. We can get the gene names from the feature ID from the object. Let's run. And we can convert it to a data frame. Let's convert it. We named it as the feature ID. We can click it. You can see here are the gene names for the joined object. I showed you before, all the mitochondria gene names started with MT hyphen. We can filter the gene names in the feature ID and only keep the gene names that started with the MT hyphen. So they are mitochondria gene names. Let's filter the gene names. If we run, you can see we generated the new data frame mito ID. We have 13 mitochondria gene names in this dataset. We can click it. You can see we only have the mitochondria gene names left in this data frame. So next we need to convert it to characters, because when we run the add feature percentage function for the mitochondria percentage DNA, we need the gene names as characters. Let's convert the gene names into characters, and we can have a look. You can see here are the 13 mitochondria gene names as the characters. Now we can use the add feature percentage function to add the data for mitochondria gene percentage. So we can set the features as mito. We know we have 13 mitochondria genes in the mito, and we create a new column in the metadata named as percentage mito. Let's add the information now. We can have a look at the cell metadata. Let's run. You can see before we have the cell ID column, they are the barcode for each cell, and we have the list ID because we joined both object AML and IPF. So in the list ID, we know which cell comes from which dataset. And now you can see we have the third column, percentage mitochondria genes. So now we can filter the object by percentage mitochondria genes. We can set the threshold as 5% to filter the dataset. Let's filter the object and only keep cells have mitochondria percentage gene below 5%. We can do it now. So I showed you the cell metadata before. We have 13,610 cells. We can have a look at the cell metadata again. You can see now we have 13,609 cells. We only filtered out one cell. And also we can have a look at the feature data, because later we are going to add the statistics for the cell metadata and the feature metadata. We can have a look at the feature data now. You can see in the feature data at the moment we only have one column, feature ID, they are the gene names. So now we can add the statistics. So it is done. We can have a look at the cell metadata again. You can see we added 3 more columns in the cell metadata. We added the feature numbers, percentage of feature numbers, and we added the total expression in each cell. So the data from here are the same as N features RNA and N count RNA when we use the SURAT package to analyze the single-cell RNA sequencing data. And we can have a look at the feature data again. You can see we added 5 more columns in. You can see in this column, we have the information about the total cells that express this gene, and the percentage of cells against the total cells, and the total expression, mean expression, and the detected mean expression for each gene. So we finished the second step to pre-process the dataset. Now we can perform dimensional reduction and data integration. So first we need to calculate the highly variable features. Then we can use those highly variable features to perform dimensional reduction. So we are going to use the calculator HVF function to identify the highly variable features. Okay, you can see we identified the highly variable features. Giotto package divided all the cells into 20 groups and identified the highly variable genes in each group. The highly variable genes were labeled as yellow color in each group, and in the metadata, if it is a highly variable gene, it will be labeled as S. We can have a look at the feature metadata. You can see we added a new column HVF. You can see those genes are not highly variable genes by the analysis. So now we can use the highly variable genes to run PCA. We can run, you can see with the default setting, Giotto package detect the HVF column in the feature metadata and use the highly variable genes to perform the principal component analysis. So in this video, the main purpose is to show you the data integration. If you watched my first video for the standard workflow, you will see those steps are almost the same. But for today's demonstration, after PCA, we can run the Harmony data integration before we run the next step for UMAP. So let's run the wrap function from the Giotto package to perform the Harmony method data integration. We can run. You can see it is very quick. We finished the data integration using the Harmony method. Now we can use the script plot to select the best PC for UMAP analysis. Let's generate the plot. And you can see from the plot, when the PC reaches to 13, 14, 15, then the information will be almost the same. Then from 15 to 30, if we include those PCs, we're probably not going to add more information. So we can use PC1 to PC14 for UMAP analysis. You can see here we set the dimensions to use as 1 to 14. Because we performed the Harmony data integration, we can set the dim reduction name as Harmony and also dim reduction to use as Harmony as well. We give a name as UMAP Harmony. Normally, if you don't set it here, it will be just UMAP. So now we can use the settings here to run UMAP. So we finished the dimensional reduction analysis and also the data integration in step 3. Now we can move on to step 4 for cell clustering. First, we need to create the nearest network. We are going to use the default method, share the nearest network. This is a similar step in thread package, find the neighbors. You can see again here we use the dim reduction to use the Harmony, dim reduction name as Harmony as well, the name as NN Harmony, and the dimensions to use is 1 to 14. So we can create the nearest network now. In next step, we can use the maiden method to find the cell clusters. And we named the cell clusters as maiden and use resolution as 0.1. You can change the resolution. If you increase the resolution, you will identify more cell clusters. Let's run the latent method now to identify the cell clusters. Following the latent method analysis, we can have a look at the cell metadata now. You can see in the cell metadata, we added a new column named as latent to show the cell cluster number for each cell in this dataset. So now we can use the plot UMAP function to look at the cell clusters. We can count the cell cluster by their cluster number in the latent column. Let's run. So you can see we have 12 clusters in the joined Giotto object. We can use the feature plot with the classic mark genes for human lung. EPCAM labels epithelial cells, PTPRC labels immune cells, CLDN5 labels endothelial cells, and collagen 1A2 labels mesenchymal cells. We know both datasets were purified mesenchymal cells. Let's run. You can see, as I showed you in the standard workflow, we have some small cell clusters as contamination during the cell sorting process, but the large cell clusters are mesenchymal cells. You can see they show high expression of collagen 1A2. Because we perform the data integration for the healthy non-sequencing data and the IPF sequencing data, I showed you in the metadata, each cells were labeled by AML or IPF in the list ID column. We can use the group by function to separate the cells for AML and IPF on UMAP visualization. Let's run the dimplot 2D function with the settings here. We can run. Let's zoom in. You can see different cell types from the healthy human lung and the IPF human lung are integrated very well. You can see in the IPF lung, there are many cells in this cell cluster because those cells are fibroblasts. In IPF lung, they were differentiated into myofibroblasts. So we finish the data integration. In the last step, I want to show you how to save the Giotto object. You can use the save Giotto function. The object name will be AML IPF. Then you set a files directory. You can see I set into lung folder in my computer, and we can generate a new folder name as AML IPF. So let's save the data we analyzed today. Let's run. You can see R start to write the feature information, spatial information, and the image information. Because this is the function also created to save the spatial data. But actually, we don't have the spatial information and the image information in today's analysis. Okay, we saved the Giotto object. If we go to my lung folder, you can see we generated a new folder. The folder name is AML IPF. If we click this folder, you can see we saved the Giotto object as the RDS object. So next time when you want to look at the data again, you can just use the load Giotto function to load the analyzed object into R. So data integration is very important for single-cell RNA sequencing data analysis. If you have more than one dataset, It can get rid of batch effect, technical effect. You can see the whole process for data integration is very similar to the standard workflow I showed you in the first video. The difference here is that in the beginning we load more than one data set. Then after that, we need to join the data set to one Giotto object. After that, the pre-process step is the same. The only step we add in is the data integration step. We use the harmony method just after PCA. So you can see if you use the harmony method to integrate your dataset, it is a very easy step to perform the analysis. Okay, I'm going to stop from here for today's demonstration. I hope this video can help your data analysis. If you like my video tutorial, please subscribe my channel and also share my videos with your friends. Thank you.